Hello, welcome to Biology, the scientific study of life. Prokaryotes have both harmful and beneficial impacts on humans. While the best known prokaryotes tend to be those that cause illness in humans, these pathogens represent only a small fraction of prokaryotic species. Many other prokaryotes have positive interactions with humans, even serving as essential tools in agriculture and industry. Pathogenic prokaryotes. The prokaryotic species that are human parasites deserve their negative reputation. All told, prokaryotes cause about half of all human diseases. Between 2 and 3 million people a year die of the lung disease tuberculosis, which is caused by the Bacillus microbacterium tuberculosis, while another 2 million die from various diarrheal diseases caused by other prokaryotes in the United States. The most widespread best carried disease is Lyme disease, caused by a bacterium carried by ticks that live on deer and field mice. Lyme disease can produce debilitating arthritis, heart disease, and nervous disorders if untreated. Pathogenic prokaryotes usually cause illness by producing poisons, which are classified as exotoxins or endotoxins. Exotoxins are proteins secreted by prokaryotes. Cholera, a dangerous diarrheal disease, is caused by an exotoxin released by the Proteobacterium vibrio cholera. The exotoxin stimulates intestinal cells to release chloride ions into the gut, and water flows by osmosis. Exotoxins can produce disease even if the prokaryotes that manufacture them are not present. For example, the fatal disease botulism is caused by botulinum toxin, an exotoxin secreted by the gram-positive bacterium Clostridium botulinum as a ferments and probably canned foods. Endotoxins are lipopolysaccharides, components of the outer membrane of gram-negative bacteria. In contrast, in exotoxins, endotoxins are released only when the bacteria die and their cell walls break down. Examples of endotoxins producing bacteria include nearly all species in the genus Salmonella, which are not normally present in healthy animals. Salmonella typhi causes typhoid fever, and several other Salmonella species, some of which are frequently found in poultry, cause food poisoning. Since the 9th century, improvements in sanitation in the developed world have greatly reduced the threat of pathogenic prokaryotes. Antibiotics have saved a great many lives and reduced in incidence of diseases. However, resistance to antibiotic is currently evolved in many strains of prokaryotes. As we read earlier, the rapid reproduction of prokaryotes enables genes conferring resistance to multiply quickly throughout prokaryotic populations as a result of natural selection, and these genes can spread to other species by horizontal gene transfer. Horizontal gene transfer can also spread genes associated with virulence, turning normally harmless prokaryotes into fatal pathogens. E. coli, for instance, is ordinarily a harmless symbiont in the human intestines, but pathogenic strains that cause bloody diarrhea have emerged. One of the most dangerous strains, called O157-H7, first came to the attention of microbiologists in 1982. Today, it is a global threat. In the United States alone, there are 7,000 75,000 causes of O157 to H7 infection per year, often from contaminated beef. In 21, an international team of scientists sequenced the genome of O157 to H7 and compared it with the genome of harmless strain of E. coli called K12. They discovered that 1,387 out of the 5,416 genes in O157 to H7 have no counterpart in K12. These 1,387 genes must have been incorporated into the genome of O157 to H7 through horizontal gene transfer, most likely through the action of bacteriophages. Many of the imported genes are associated with the pathogenic bacterium's invasion of its host. For example, some genes code for exotoxins that enable O157 to H7 to attach itself the intestinal wall and extract nutrients. Pathogenic prokaryotes pose a potential threat as weapons of bioterrorism. In October 21, endospores of Bacillus anthracis, the bacterium that causes anthrax, were found in envelopes mailed to members of news media, and the USA Senate 18 people developed causes of anthrax and 5 died. Other prokaryotes that could be candidates as weapons include C. Botulinum and Yersinia pestis, which causes a plague. The threat has stimulated intense research on pathogenic prokaryotic species. In May 23, scientists at the Institute for Genomic Research in Maryland published the complete genome of the strain of B. anthracis that had been used in the October 21 attack in the hope of developing new vaccines and antibiotics. Prokaryotes in research and technology. On a positive note, we reap many benefits from the metabolic capabilities of prokaryotes. For example, humans have long used bacteria to convert milk to cheese and yogurt. In recent years, our greater understanding of prokaryotes had led to an explosion of new applications in biotechnology, the use of E. coli in gene cloning and of agrobacterium tomofacent in producing transgenic plants are two examples. Prokaryotes are the principal agent in bioremediation, the use of organisms to remove pollutants from soil, air, or water, organic matter in sewage, converting it to material that can be used as landfill or fertilizer after chemical sterilization. Other bioremediation applications include breaking down radioactive waste and cleaning up oil spills. In the mining industry, prokaryotes help recover metals from ores. Bacteria assist in extracting over 30 billion kilograms of copper from copper sulfides each year, harnessing other prokaryotes that can extract gold from ore. And on factory in the African nation of Ghana processes 1 million kilograms of gold concentrate a day, about half of Ghana's foreign exchange. Through genetic engineering, humans can now modify prokaryotes to produce vitamins, antibiotics, hormones, and other products. One of the most radical ideas for modifying prokaryotes has come from Greg Venture, one of the leaders of the Human Genome Project, who has announced that he and his colleagues are attempting to build synthetic chromosomes for prokaryotes, in fact, producing entirely new species from scratch. Venture hopes to design prokaryotes that can perform specific tasks, such as producing large amounts of hydrogen to reduce dependence on fossil fuels. The usefulness of prokaryotes is largely derived from their diverse forms of nutrition and metabolism. All this metabolic versatility evolved prior to the appearance of the structural novelties that heralded the evolution of eukaryotic organisms, to the topic of the remainder of this unit.